The overall goal of the following experiment is to measure receptor-ligand interaction kinetics when both molecules are anchored on the surfaces of the interacting cells. This is achieved by first functionalizing the red blood cells, which are the adhesion sensors, with ligands, and preparing receptor-bearing cells. As a second step, the micropipette chamber is assembled and adhesion tests are performed to record the adhesion events. Next, the adhesion probability versus cells contact time curve is plotted and fitted with the model in order to estimate the kinetics parameters. Results are obtained that show two-dimensional effective binding affinity and off rate estimated from the data.
So where do we get the idea of this assay? People study receptor-ligand interaction, mostly measure rupture forces, and we are doing the same thing. But one day we thought, could we just measure the binary outcome, 0 for no adhesion and 1 for adhesion, and derive the measurement for kinetics from those 0 and 1 measurements? To show the micropipette adhesion frequency assay, Dr. Veronika Zarnitsyna, a research scientist of my laboratory, will show the experiment to you.
Red blood cells isolated from whole blood are biotinylated for the adhesion frequency assay. To begin this procedure, centrifuge the tube containing isolated red blood cells. Aliquot 10 microliters of red blood cell pellet into each of several vials and add the